Somehow I seem to have lost my CCMV image up here. I'm not quite sure how that happened. No? Somebody decided it was copyrighted or something, which is probably why the DVD recording last time didn't work. So I very carefully ran the DVD off my computer so you could all see it here, and it was on the other screen. And then I looked at my screen recording, and it was gone. Well, actually, it wasn't gone. It just had this really strange checkerboard. So I suspect it's some kind of copy protection thing. Has anybody managed to find the DVD in the library? Has anybody tried? Okay. Come on now. You got like, how many hours until the midterm? It's like tons of time. Should be no problem at all. So my apologies about that. I thought I would be able to record it. I looked online, YouTube, etc., to see if there were any copies that happened to have appeared there. It doesn't look like there are any. That's the main thing as far as that DVD is concerned. Really has to do with the packaging aspects that we looked at right at the end of class, so how the terminase is binding to the concatenameric genomes, and I think it's way better shown in those animations than what I was able to sort of hand wave about in class in these static images. So that was sort of the main thing. And if you do, unlikely event, go and try and check this out in the library, look at the overview and highlights, which is the first part of the menu, and then the last part, which was the, again, packaging and escape from the cell in terms of those enzymes. So again, this is my normal review pattern, but first we have to have our clicker questions because everybody loves clicker questions, right? So first clicker question is, a bacterium with a provirus in its genome is called temperate, a lysogen, E. coli, a host, restriction modified. So 53 have voted and nobody's changing their mind. It's a good thing. Yay, everybody's happy, including me. So what's temperate? The phage is temperate, exactly. If it can undergo lysogeny, but a bacterium with a provirus in it is a lysogen, assuming it's a functional provirus. There are lots of non-functional proviruses that people find in genomes, which we haven't talked about, but it's true basically all throughout the bacterial kingdom as far as we know. If I'd asked about a bacterium with Lambda prophage in it, that would have been E. coli. And a host, actually it's probably not a host anymore because most of the time you have superinfection immunity, and certainly that's true for Lambda. And restriction modified, you had to have something to come up with for E, right? But the restriction modification, in fact, could be a reason that you're not going to have that provirus, or that the provirus actually can't get out. Because once a provirus is in the genome, it's going to get modified just like the host genome gets modified. So the way that restriction endonucleases work is that they will cleave 99% of the time non-methylated DNA. And so if the virus comes inside the cell and it's non-methylated, it'll be chopped up by restriction endonucleases. Once you're a provirus, of course, you're in the genome and you get methylated by the normal cellular methylation processes. So yeah, there is method to the madness on some of these questions here. So next clicker question, lytic reproduction in Lambda bacteriophage, well, I should start, is due to the activity of transcriptional repressors, transcriptional activators, transcriptional anti-terminators, A and C or B and C. Eight, 7. Oops, let's show the results. Yes? Okay. Most people think D. Fortunately, I think D as well. And by the time you put A and C together, that actually comes up with D as well. So we're in pretty good shape. Yes, lytic reproduction, but really important, this is lytic reproduction. Lysogenic reproduction actually has to do with transcriptional activators as well as transcriptional repressors, which brings me to my centerfold here from the book that you can check out if the DVD is not enough information for you. Lambda 2, basically more information than probably most people ever want to know about Lambda. But it gives you an idea that it really is very much a paradigm for understanding molecular biology in general. A lot because of these transcriptional regulators, which are involved in the whole process. So what's the transcriptional repressor that's most important for lytic growth? Okay, is that important for lytic growth? What happens when you don't have C1? Mutant. You still get lytic growth. So what repressor is important for lytic growth? I'll let you think about it. Transcriptional activator, C1 can serve as an activator, but that's, again, really important for lysogenic reproduction. Transcriptional anti-repression, I heard lots of people saying this already. So what are the anti-terminators? N and Q, exactly. And so the answer is D, but it's mostly the anti-termination, which is critical for this. So select our answers. And our next question, which kind of leads into this, if a bacteriophage lambda had an inactivating mutation in CRO, it would only reproduce lytically, only reproduce lysogenically, reproduce normally, not be inducible, not be able to replicate. 10. It's time to start guessing. Okay, so what's CRO do? So it blocks what aspect of C1? Okay, not really binding to the origin of replication. Origin of replication is over with O and P. So that has very little to do with that. What promoter is it associated with? So PR, but what else? And so what's CRO stand for? Control of repressor and other things. What do PR and PL have to do with repressor? Nothing. So how is it controlling C1? Which does, yes. And what does that then block?
Expression of the C1 protein.
From which promoter?
PRM.
PRM. So, and that promoter is for what? Repressor maintenance. So, once you've already made C1, it's actually not regulated by CRO. So, if you get rid of CRO completely, you can still make C1. So you can still undergo lysogeny, but you can also reproduce lytically, because it's going to still be a possibility which one. Turns out it actually lyses more than going through lysogeny, but it does both. And that's partly why it's not like a clear mutant. So if you would expect, we already talked about our clear mutants, C1, C2, C3, mutations to any of those, it's always going to go lytic. But CRO, okay, it's involved in regulation of these things, but they would have found it as one of these clear plaque mutants, which they didn't. And so that gives you an idea. This was a much, much, much more difficult question than any of the other ones. I think probably any of the ones we've had so far this term. But it requires you thinking a lot about so it does reproduce normally. It's inducible just fine, because if you are replicating lysogenically, you've got C1, that'll get cleaved. That has nothing to do with CRO, and replication also works. So that being said, that is a nice reminder that I'm going to normalize to the high score. And that means that questions like that, for instance, if no one had gotten, although one person got it, you got to kick them out, purge them from the classroom. That question basically wouldn't count in terms on the exam. Yeah, well, that's another possibility. You never know. So maybe you don't want people guessing on the exam. So this is basically just a really quick review. Hopefully we've gone over the important aspects here. Just one thing I wanted to mention again was the plaques, and we'll talk a little bit more about plaques a little bit later on. This particular plate is from a C1 mutant, because all of the plaques here are nice and clear, and that means that lytic replication has happened. If this was a wild type Lambda, all of these plaques would have a little bit of growth in them, and that would be due to the lysogens which were in that particular set of cells there that were being infected by the virus. And since once you're infected with Lambda, you're producing C1 because of PRM, and the really complicated question I could have asked, even worse than that last one, would be superinfection immunity, because it turns out that CRO mutants are not superinfection immune. Because when you produce lots of C1, then any new virus that comes in will be blocked. However, if you're using really small amounts of C1, that could actually be replicating lytically. But we didn't want to get quite that far. So again, these are clear plaques because you don't have lysogenic growth going on. And turbid would be you have a little bit of growth going on here in those plaques. And I guess I didn't explain that terribly well last time. Okay, any other questions about Lambda? If you do, come borrow the book. So this, again, these are just review from what we've had since the beginning of the term. If you take nothing else away from this class, I hope that you get the concept that a virion is not a virus. And yeah, I know I can't say, okay, fine, you got that concept, you got an A for the class, you're good. No, unfortunately, it doesn't work that well. Sorry. But virions are basically bags of nucleic acid. We'll talk a lot more about some of these nucleocapsids on the inside of enveloped viruses in the next part of the term. But basically, this is the bag, that's nucleic acid on the inside. And again, that's the extracellular form of the virus as it's replicating. Envelope proteins we've talked a little bit about in terms of entry, but we'll talk a lot more about them as we talk about some more of these viruses a little bit later on. And just to beat the dead horse, ceci n'est pas un virus, this is just the virion. It's not the virus itself. So that being said, viruses infect everything. Anybody found an organism that's not been infected by viruses for extra credit? It's probably not worth your time. Because as far as we know, all of these organisms have viruses associated with them. Many times those are actually proviruses, which have been integrated into the genomes, particularly for bacteria and archaea. Certainly for us, we've got lots and lots and lots of proviruses in our genome. We'll take a look at that in just a second. The other thing about these viruses is that they're probably extremely old, and I talked about some of my ideas about phylogeny of some of these viruses, and clearly really, really important for evolutionary processes. Now, if you're a bag of nucleic acid, that nucleic acid can be a gene, and so you're moving stuff around all the time. Massive numbers, again, maybe the second, I think, most important thing to take away from this class is that there are absolutely ridiculous numbers. Of course, in this case, these are all virions that we're looking at. A lot of these cells here in the background, the bacteria and archaea, are probably part of the viral life cycle too, because they're probably infected. So virions, absolutely ridiculous numbers, but also lots and lots more cells. Mentioned before, our own genome, of course, has 8% of clearly retroviral-like elements that are present inside the genome. And again, arguably, these line and sign elements are also viral. Certainly, they replicate through a reverse transcription mechanism, which is really similar to the retroviruses. So my personal feeling is that 40-ish% of the genome is viral, at least virally derived. And of course, this puny little bit over here, the 1.5%, which is actually protein coding. That's probably an oversimplification, because if you think about where the introns are, which are in these genes, those are really pretty huge. And as we mentioned in that other course, which will remain unmentionable, there seems to be transcription and regulatory elements spread really all throughout the genome of this part over here, which is unique, but also a lot of the stuff over here as well. So what that's doing is still really not completely clear. Viruses contain the most genetic diversity of anything that we know of. If you look at really well-characterized bacteriophage, 50% of the genes don't look like any other gene that's out there. In metavirome sequences, and we'll talk a little bit more about metaviromes in the next part of the class, basically you purify the virus-sized particles, or virion-sized particles, I should say, and sequence those. 90% of those sequences don't look like anything else anybody's ever seen before. So this is the dark matter of sequence space. And then some of the extremophile viruses are 95%. One of my favorite viruses here, the SSV viruses, have one of their 35 genes that are similar to other genes in databases. So it's a ridiculously small amount of virus sequence diversity that we know about. And this is just an absolute estimate here. want to spend a little time talking about plaque assays. By the way, stop me at any point if you'd like me to explain any of this in more detail.
I have a question.
Yeah, sure.
Should we remember these gee-whiz facts for the quiz? Like
The quiz on Monday?
Yeah, not just the overall, just the...
Yeah, so I would, maybe we'll back up real quick to that, that slide. I would say, remembering that, 90% of sequences that you find in viruses are usually unknown. That would be reasonable. That it's 91% in this paper or 95% in these extreme viruses. No,
Okay.
I wouldn't. 10 to the 6th to 10 to the 7th per mil in ocean waters, I think that's reasonable. 10 to the 31, or that it's 200 million light years when you stretch them end to end, I don't think is reasonable. So it's sort of the basic facts, as it were, at least the experimental data,
Okay.
I would say. But the massive extrapolations, no.
Okay. Perfect.
Okay, other questions about the big picture stuff, the trivia questions that I asked at the Science Pub the other day. So, yeah, plaque assays. And so, again, this is a nice example of some plaques here, clearly clear plaques, pun not intended. These, in fact, come from Bacteriophage T4, which just replicates lytically. This plaque here is probably millions to tens of millions of viruses, being virions and infected cells. But that comes from originally one virion that was infectious. And so the way that you get this, and again, you can just back calculate, you count the number of plaques on your lawn, you can back calculate to the number of bacteriophage that you started with. Same thing is true for a lot of the mammalian viruses, anything that you can study by a plaque assay. So you have a dilution that you start with, you mix it together with your host, put it in something which is going to immobilize the host and the virus, so whatever infection happens can't get very far. Mix them together and just let them sit. And this sitting process is multiple rounds of replication. And so that's really important because otherwise you would never see the plaque, because you have to have multiple rounds of infection to actually see that plaque. The individual virion you're never going to see in this case. So are people clear on plaque assays? This is, again, a pretty important concept and how you could go about doing these. Mostly for these plaque assays, so you can do one-step growth curves. And the one-step growth curve is getting back to that same issue we just talked about with the plaque assays. And that is that it's really, really hard to study one virion and one virus infection. So what you want to do is study multiple infections that are happening all at the same time. And so basically by taking the mean of that, it gives you an idea how each individual virus is going through its replication cycle. So you have to have as synchronized infection as you possibly can, which means you're using a very high MOI, high multiplicity of infection, and just statistically that means you'll have at least one genome that gets into all of the hosts that you're looking at. And so that's here, PFUs per cell. Classic case here, you start out with a MOI of 10. So 10. Then infectious viruses goes down to, in this case, 1. That's due to the infected cells. So again, if you're thinking about the viruses, if you do this assay, each one of the cells that you have in there is going to be a virus because it's been infected. Usually here you do a dilution or a wash just to get rid of any further infections that take place. And then you just follow with time. And now we're looking just at the black line on here. This is just the plaque assays. So over about a little over a day and a half, that's how long it takes for one replication cycle to take place. So if you're thinking about your plaque assays, these are probably going to take a week or two to actually get plaques. So you do the experiment, you collect all your samples, and you do the plaque assays and eventually get your data probably weeks later. It's easier to look at some of these other things because they don't take as long. And if you look at messenger RNA, for instance, really nice in this brown dashed line. Here you see a bump in viral messenger RNA. Those are your early genes that are being expressed. And then you see a really big pickup in terms of that RNA. And that's because you're also making a lot more of the viral DNA, which is this continuous line right here, or the genome. In this particular case, actually an RNA virus. But the genome sequence, oh no, it has a DNA virus. This is the viral DNA. But you replicate your genome, and then from that genome, you're going to be making your late genes. And those late genes then are the ones which are giving you much more RNA. Protein production, in this case, T antigen. This is just proteins in general. Sometimes you'll see a bump at the beginning, but very often the proteins you make at the beginning, they're going to be enzymatic proteins. So you actually don't need very much of them at all. So it can be really hard to detect. And so you often don't see much of a bump unless a lot of the structural proteins that you see, which are really taking off a little bit later on. So you can get huge amounts of data from these one-step growth curves. Tells you a lot about how the virus is replicating. We talked a little bit about enveloped versus non-enveloped that you can get from this. You can also get burst size, how many virions are produced per infected cell. And as you can see here, this is a really pretty humongous one. It's on the order of 700 or 800 in terms of your burst size for any given cell. So people clear on the one-step growth curves. Again, this is a pretty fundamental concept in virology. If you don't have a Plaque assay, it makes these really hard to do. And so most of the people who are studying standard viruses have established a Plaque assay. If you find a new virus, one of the really important things to do is establish a Plaque assay and do a one-step growth curve. Yeah?
I'm assuming there's sort of a correlation between the virion size and like the maximum burst size. Like the ones that would have 1,000 virions per cell would be smaller.
So yeah, the question again, just for the tape, assume that larger burst size are probably going to be smaller virions. That's true if you assume that the host is always going to be the same size. And it turns out that there are certainly hosts that are a lot bigger. And so the example that I always like to use for that is virus, which is one of the largest viruses known. It infects amoeba, which are actually pretty large cells. And so those also have pretty large burst sizes. So relative to your host size, then you're going to have a higher burst size. They're usually going to have smaller particles. So that's a reasonable thing. But again, it's going to depend on your host size. Other questions about this aspect of things? Okay. 6 different kinds of viruses. I don't expect you to remember all 6, but that there are 6, and you could probably put them together. I wouldn't say what's class 6, what's class 4, what's class 1. I can't remember these things, but that there are 6, and 6 different ways of getting yourself to your messenger RNA, I think is reasonable. Again, it's all about the messenger RNA because it's always cellular translation. And so it's how you get to that cellular translation process. And the way that these are all split up mostly has to do with what kind of nucleic acid is inside your virion. The exception are these reverse transcriptase viruses down here at the bottom, which actually can package multiple different things, but are still classified as reverse transcriptase-like viruses. But single-stranded DNA viruses, double-stranded DNA viruses, single-stranded RNA, positive or negative, and double-stranded RNA. So those are your 6 basic classes of viruses. So far, we've just talked about single-stranded RNA, single-stranded DNA, and double-stranded DNA viruses. We'll talk about the negative strand RNAs and these retroviruses. May or may not have a chance to talk about the double-stranded RNA viruses. It turns out that the double-stranded RNA viruses almost always replicate very similarly to the single-stranded negative strand RNA viruses. And probably, again, just important concepts to get from here. If you're a negative strand, or for that matter, double-stranded RNA in your virion, you have to bring a RNA-dependent RNA polymerase with you because you can't make messenger RNA. There's no cellular protein which will make a copy of that RNA that comes in at least not a big enough piece that can serve as a messenger RNA. So there you absolutely require a viral protein to come in. The same thing is true of these retroviruses here. They require reverse transcriptase that has to come with them. Yes?
It says they have positive single-strand RNA twice.
Yes, and so the difference down here is the reverse transcriptase. And so this really could be single-stranded RNA, it could be double-stranded DNA, it could be single-stranded DNA, and it turns out that all of these retroviruses that go through the reverse transcriptase process can have all of these different things in terms of what they're packaging. So this is sort of the exception as opposed to what's being packaged. The ones that most people study, i.e. HIV and friends, are these positive strand, single-stranded RNA that's packaged. But you certainly have the double-stranded DNA as well that's packaged and still goes through this reverse transcription process. Okay, that's, again, we'll talk a lot more about each of these as we move along through here. I brought my wonderful icosahedrally symmetric particle with me today, so if you're interested in checking this out a little bit later on. In general, and again, exceptions for things like Mimi virus and some of these really giant viruses, smaller is better, and this really has to do with the economy, genetic economy. It takes a lot of your genome to encode a capsid protein. So the fewer of those capsid/structural proteins that you need to encode, the smaller, more efficient you can be, and potentially, getting back to your question, burst size-wise. You can have a much bigger burst size, and generally, bigger burst size is going to be better in terms of passing along your genes. So the reproductive strategy, generally, again, smaller is better. How do you get smaller? Helical is, in fact, the simplest way to do that. But the problem with helical symmetry is you have the ends, and you have to deal somehow with the ends. Way to avoid dealing with ends, of course, is to not have ends and have some kind of spherical particle, or at least as close to a spherical particle as you can get. And that, of course, is icosahedral symmetry. Icosahedral symmetry is really easy if you just have 60 capsid proteins all fit together in a nice icosahedron. Those usually don't work. To get even the capsid protein gene, let alone everything else that you need to package to be inside your virion. So usually these icosahedrally symmetric virions have extra copies of, in many cases, exactly the same coat protein that get put into the particle in addition to the 5 coat proteins which are around each 5-fold axis of symmetry. And so this is the whole concept of quasiequivalence, where you have exactly the same protein that either 6 of them associate with each other or 5 of them associate with each other. And in that process, you can put together lots and lots and lots of these 6s that associate with each other in very similar ways to the way they did here with just 5s. And so that's your quasiequivalence. And you can get T numbers by going from 5-fold axis to 5-fold axis via these hexameric subunits that you've put in between. So h squared plus hk plus k squared, that is something I would expect you to remember. And if I gave you something like this in the exam's not written yet, so I don't know exactly. So anything I say now is pure speculation. But if I were to give you a icosahedral particle, I would give you something like this. I wouldn't give you a cryo-EM to look at, because I usually have a lot of trouble finding those. But here, go from this 5-fold axis of that 5-fold axis of symmetry via where you have your hexamers. You go 1 to the purple ones. From the purple ones, change direction, go another. Back to the 5-fold axis of symmetry gives you 1 squared plus 1 times 1 plus 1 squared equals 3. On the other hand, up here in the upper right-hand side, 5-fold axis up at the top, another 5-fold axis down here at the bottom, 2 5-fold axes in the middle. To get from 5-fold axis to 5-fold axis, you need to go to 6-fold, but you keep going in the same direction. to get to your five-fold axis of symmetry. This is also quasi-symmetric interactions. These guys are still interacting with each other, but they're in slightly different ways than you have down here. But that's a H is equal to 2, K is equal to 0, so it's a T equals 4 particle. So in this way, you can build up larger and larger structures just by using one or a very small number of capsid proteins. And as we mentioned before, even things like virus, these not quite the biggest viruses anymore, but pretty close, have these icosahedrali symmetric structures and use very few different coat proteins, but many, many copies of those individual coat proteins. Questions on virus structure, quasiequivalence, etc.? I don't expect you to remember the derivation of these numbers. If you want to look at Euler's theorem, more than happy to, not in this class. So one thing that I think is, so if I were to give you, again, back up a little bit, question on an exam for something like this, if you had a cryo-electron microscope structure, I would give you the hexameric subunits here that you could use to follow along. So this is one of my favorite viruses, probably my second most favorite virus, STIV, which is a T equals 31, 5-fold to 5-fold, 1, 2, 3, 4, 5, and 1. And the important thing here, however, has not to do with the icosahedral symmetry, but the capsid proteins and their overall structures. And it turns out that you can put the capsid proteins together in STIV with a T equals 31, basically an identical capsid protein in a bacterial virus. This one's a T equals 25 because it's just 5 along the side. Same thing is true for adenovirus. Same thing is true for PBCV1, which is an algal virus. That, it turns out, has a much larger T number, but again, the same structure of the protein. And so the idea here is that protein structure is probably a much better way of thinking about really deep phylogeny than all those processes that I brought that big black book about, the ICTV. It's great for stuff out here, viruses that are infecting various different cells, particularly animals and plants over here. Really short distances in terms of phylogeny. The ICTV comparisons are really useful. In terms of deep phylogeny, we really have to use different tools. Yeah?
Just as a problem-solving mechanism for
Yeah.
figuring out the T number, so would you recommend drawing a straight line between the two pentamers? And then it seems like sometimes the hexagons take you in a perfect straight line there, or sometimes they veer off to the side. And they veer off the side, get as close as you can to the next 5, and then just cut over.
Right. Yeah, that's a really good way to think about it. Yeah. So the idea here is, and sorry again to repeat for the recording, is what's a good way to go about doing one of these things? If I were to give you a question like this, is basically find the straight line like the red lines on this figure from 5-fold to 5-fold, and then find where your hexamers are. These are also known as capsimer subunits. Sometimes people call them as capsimers. And then count along those in the direction closest to the line that you've drawn. And then as soon as you get as close as you can, change direction and go one more. Or in case of a T equals 7, it's usually hop 2 and then go 2 in the other direction. So, but yeah, it's a really great strategy for looking at something like this. Yeah?
Okay, so when you're hopping, are you going, so you go to your straight line and that's like your K. And then is your H how many to that 5-fold axis of symmetry, or how many to the next straight line, like to your next K?
Okay, maybe I'm not. So why don't I actually go ahead and pull my pen up here so we can just do one real quickly. So, oops, come on. PowerPoint is wonderful in some ways and really obnoxious in others. So here's our pen. Pen, yes, pen, say pen. Is it not talking to my pen? Do I need to go over here, do it this way? Pen structure, yes, there we go. Pen color, red. Good, okay, so let's just take this one right here in the middle. So here's our 5-fold axis symmetry, and you've just drawn your red line between the two here. We'll start here, we'll go 1, 2, 3, 4, 5. If we were to keep going, we're going to go past that 5-fold axis of symmetry, so that's no good. Now we need to change direction, and we go 1. And so it would be H of 5, K of 1. Let's look at the bacterial one over here. Here, 5-fold axis of symmetry, 5-fold axis of symmetry. Hop between the two. If my mouse reappears here, there we go. So we go 1, 2, 3, 4, 5, and we don't need to change direction. Does that make more sense?
And do you count when you land on the pentamer, you don't count that as part of your, on that last example?
No, you do. So that's, yeah. So this is going to be t equals 25. So you do a callus. We'll do over here because it's actually exactly the same thing. So here, so you start at the beginning. This will be 1, 2, 3, 4, 5. Happy? Unhappy? Confused? Never thought you'd have to do geometry in a virology class? Okay, so talk a little bit about virus entry. This slide is basically as a reminder that most of the viral receptors, which of course are cellular proteins that did not evolve to bind to viruses, these are things that are sticky, that stick to stuff on the outside of the cell. So CD4, these poliovirus receptors, Coxsackie adenovirus receptors, integrins, they're all things on the outside of the cell which bind to things. And what kind of structures do they often have? Immunoglobulin-like folds, because immunoglobulins are also really good at binding things. But again, it's not just those. You have some of these other proteins as well. And we'll talk much more about these when we talk about the individual viruses a little bit later on. Hopefully, I haven't said this too many times, but in terms of virus entry, this is the figure that you really should be studying. All of the different processes whereby you can get your virus genome inside the host. Let's start just with, let's see, this one doesn't have the viruses which are just injecting themselves directly. So you could have a bacteriophage, so it's not complete. Bacteriophage, it would be injecting its genome right here across the membrane. But usually you'll have incorporation into an endosome, like you have with the influenza virus, then a structural change, and get fusion. You can also have fusion that happens at the plasma membrane. In many, many cases, you want to get your virus genome to the nucleus, and that's because that's where the cell is replicating things. And so if you want to replicate your DNA, you want to replicate yourself in the nucleus. And there are, again, lots of nice animations that I didn't put in here, but they're all linked in my various lectures. Probably the most, I think, I don't know what the right term is, but spectacular is maybe a good one. Of these animations are how you get membrane fusion taking place. And the real key to membrane fusion is some kind of change that's happened when you have association of your receptor and your viral receptor binding protein. And that change is almost always a structural change in that particular protein. And so in the case of influenza hemagglutinin, you have binding to the receptor on the outside of the cell, then conformational changes in the fusion peptide, which then has further conformational changes which pull the two membranes together, eventually getting them to fuse, and the virus genome, although usually it's virus genome plus proteins, which are then released in that process. So that's fusion, peptides. Also wanted to mention really briefly this process in polioviruses. It's not just membrane fusion, but conformational changes in general of viruses. Even the naked viruses, like poliovirus, undergo a conformational change. drugs that block those conformational changes are really good antiviral drugs. And so that's just what this example is supposed to be here. That you bind to, you block any of those changes that are taking place, which are absolutely critical for releasing the genome inside the host. Now I want to switch gears for the last 15 minutes or so and talk about the individual viruses that we've talked about so far and sort of the highlights or lowlights, this depends on your point of view, of each of these particular viruses. These are the single-stranded RNA bacteriophage. Single-stranded RNA is, of course, a complete misnomer because most of it's in secondary structures where it's double-stranded. And it's really those secondary structures which are most important for how the virus does what it needs to do, i.e. get, in this particular case, and true really for pretty much all viruses, differential amounts of capsid proteins, replicase proteins, and in the final case of these single-stranded RNA bacteriophage is the maturation protein and lysis proteins. So the way that this is being regulated really has to do with the availability of start codons. The capsid protein, really easy to get to that start codon. You can get lots of translation. It's only after you have lots of translation and then this translation process which disrupts some of these secondary structures that you can start to make some of the non-structural proteins, and in the case of these single-stranded RNA bacteriophage, those are replicase and lysis proteins. So they are non-structural proteins, the things that you're not going to need. The last one, at least in far as this part of the genome, is the lysis protein. That lysis protein is also made by different translation. So it's all about how that translation is being regulated. Here, it's because the ribosomes, once they've got through the coat protein, they don't always immediately dissociate. They bounce around a little bit and then can actually start at the start codon for the lysis protein. So those are the, basically the three proteins that you need, with the exception of one for all these small RNA phage. The last one's the maturation protein. This one's not dependent on translation. It's dependent on replication. And again, it also makes sense. We'll hear much more about this later on in a lot of the other viruses. The only way you make the maturation protein, which is really the last protein that you need in order to make virions, is once you've made genomes. And the new genomes, once you've made a new genome that's going to get packaged, the new genome, again, makes a secondary structure, as you have for all of these RNA viruses. But that new genome makes a specific secondary structure which allows the maturation protein to be made. If you have old genomes, it's never going to be made because you don't get translation from starting. So it's still translation, but how that translation is regulated is not due to translation per se, but it's due to production of your template, how you're going to be replicating any of these individual viruses. Okay, any questions, more questions on single-stranded RNA bacteriophage, single-stranded DNA viruses, or I should say single-stranded phage. We'll talk more about single-stranded DNA viruses of other organisms later on. Phyx 174, classic example, interaction with the outside of the cell, and I didn't talk about that particular entry process. If you're interested, we can talk about it later, but it won't be on the exam. Once it gets inside the cell, you've got this single-stranded DNA. Single-stranded DNA has actually similar kinds of problems to single-stranded RNA. The negative strand sort, it can't do anything until it becomes double-stranded, and that, of course, is due to an actual slight double-stranded piece of DNA which is present in all of these genomes. Once you make double-stranded DNA, which we'll look at in just a second, you end up making a number of copies of double-stranded DNA. That is your template for messenger RNA, which gets made into protein, but also is the template for rolling circle replication, which makes many copies of your positive strand, your single strand. These guys come together and they get released from the cell. Replication, again, is really due to that first step of replication, I should say, double-stranded pieces which are present in your genome, even though it's a single-stranded DNA virus. So this structure up here at the top, the hairpin structure, again, it's just complementary sequences, a lot like the RNA phage. Those complementary sequences will bind to each other, provide a binding site for cellular proteins, which can then prime replication of your genome and regular replication, RNA primers, DNA polymerase III, making double-stranded DNA. That double-stranded DNA gets replicated into single strands through the activity of this A protein, also recombinase protein. Tyrosine will come in, nick the backbone, provides a 3' OH that your cellular polymerase can extend. There's also helicase activity, which will peel off the single strand, and your DNA polymerase will just go round and round and round the genome, making multiple copies of your single-stranded piece. That single-stranded piece then comes together with capsid proteins and 2 different kinds of scaffolding proteins in order to get packaged into this procapsid structure, and it's really this assembly process with the 2 kinds of scaffolding proteins that's completely unique to Phi-X174, but the overall assembly process, putting together pentameric subunits, those pentameric subunits coming together to make icosahedral structures, is a theme we'll see also again and again as we move on through the rest of the course, because icosahedra are pretty darn good ways to package your nucleic acid. Questions on Phi-X174 and microviruses? Then we talked about bacteriophage T7, which has T = 7 icosahedral symmetry, but much more important in terms of understanding T7 and why we're using it as a nice paradigm has to do with how it's making its individual genes, and particularly the process of the T7 RNA polymerase and transcription in general. Transcription right here at the very left end, or the early part of the genome, is due to cellular polymerase. That cellular polymerase also pulls the genome inside the cell. Eventually you make the viral DNA-dependent RNA polymerase, which binds to lots of promoters here, and then leads to transcription of first your class 2 genes. Also, people talk about these early genes, making more of your genome. That's after you have the first hump that you would see on a one-step growth curve. The first hump would be here, these very early or immediate early genes, your DNA metabolism genes, and then finally very large amounts of your structural proteins at the end. All of this is due to transcriptional regulation, most of that due to your T7 RNA polymerase. And T7 RNA polymerase is fabulous because it's a single subunit DNA-dependent RNA polymerase, very specific promoters, goes really fast. And one of the things that I was thinking about doing is just presenting the size of even just the E. coli DNA-dependent RNA polymerase. It's about 6 times this size. It's just huge, relatively speaking. So it's a lot easier to work with these proteins, the single subunit RNA polymerases. Just again, this is more questions of subsequent. Yeah, sure.
With the size and the speed of the T7 polymerase, does that have a high error rate in comparison?
Yeah, so the question is, does it have a higher error rate? And the answer is yes, it does have a considerably higher error rate. And again, this is getting beyond what you need to know for the midterm, but we'll talk much more about this later. Turns out it's much more like a DNA polymerase in terms of its activity than an RNA polymerase.
Okay.
So, and those have generally lower fidelities, other than proofreading activities. So it doesn't have a proofreading activity.
Right.
So this is actually kind of getting back to questions about what do I expect you to know about any of these individual viruses. I can't remember that gene 1.3 is a DNA ligase, or that we've got inhibition of RNA polymerases, et cetera. The circle ones are the ones that we've talked more about. And importantly, here are going to be the endonucleases, which is chopping up host cell DNA. You don't want to be making any host DNA. You just want to be making viral DNA. The lysozyme, as well as getting out of the cell here, is really important for regulating the T7 polymerase. And that these viruses, the T7 virus in particular, and Lambda true as well, have a lot of their own enzymes. So DNA polymerase. I would not remember that these are genes 3, 3.5, and 5. I would remember that it's got most of these enzymes that you need. It's got its own DNA polymerase. It's got its own RNA polymerase. It's got things to get rid of host DNA, et cetera. So those are the things that I would remember as far as T7 is concerned. Any questions about T7? Otherwise, we can move on to page 95 of this. So we basically kind of covered this already when we talked about our clicker questions earlier on today. Through lytic growth, that's all about your anti-termination from N and to some extent repression of the, I guess we don't even have the PRM promoter in here, of the C1 promoter. So repression and anti-termination, which is really critical in terms of getting lytic growth. And lytic growth is pretty much the standard for Lambda. If under normal growth conditions, you're going to get lytic growth, and that's why you get plaques. If everything was lysogenic, you'd never even see plaques because you wouldn't have any lytic growth at all. There's a small amount of lysogenic growth that happens, and part of the reason for that, why you get a turbid plaque, why in the middle stuff actually still grows, is because the going's pretty darn bad for the cells which are there, because most of them are lysing because they've been virus infected. And so the host is unhappy in a turbid plaque because everyone else is getting killed off by this lysis process. And the way that you can go through this lysogenic process is activation of the C1 protein being a repressor for the lytic genes, but also an activator for its own genes. And so that's why repressors in quotes, because it's also serving as an activator of its own genes. So it's serving as both transcriptional activator and repressor. If you make enough of the C1 protein, I think I didn't sort of skimmed over this when we had our lecture before, it binds to not only the PR and PL promoters, but also makes this secondary structure which completely shuts down any kind of transcription, even of C1. So if you get to a certain level of C1, you don't make any more because you don't need to make any more. And so that really shuts everything down here completely. We talked a little bit about the genetic switch, how this happens, again, having to do with CRO. And in the absence of CRO here, you see that you have a very small amount of the PRM which is being made, and PR is on until you get enough of your Lambda C1. So it's all about how much CRO you have versus Lambda C1, but you're still going to have some of that C1 in order to be able to go through the process. The last thing that I wanted to talk about that I think I did a really crummy job of explaining before has to do with INT regulation. Now, the INT gene is what's going to be expressing the integrase protein, which, if you're going through the lysogenic process, takes the Lambda genome and inserts it into the host genome. You, of course, only need this if you're going through lysogeny. If you have N anti-termination, are you going through lysogeny? No, you're not going through lysogeny at all. You're going through the lytic process. So in the case of N anti-termination, there's a terminator here, which in the lack of anti-termination, terminators here, it makes this really stable secondary structure, which is actually not a substrate for RNase 3. And I think I misspoke about that last time. So this is not a substrate for RNase 3. This RNA is stable. It's being expressed from the pINT promoter, which is way downstream relative to PL. That promoter is stimulated by C2. So the protein which is also stimulating the original expression of C1. So stimulated by that, but you don't have any N around, so you terminate basically here at, well, right after 2 in this structure. Right after 2, you get this 2-3 structure. 4 is not base pairing with 1 because 1's never made, because you don't actually go past here. If you have this base pairing interaction between 1 and 4, that gets cleaved by RNase 3, and that chews up this end of that RNA, so then it can no longer be expressing the int protein. So in lysis, you don't need int, so you may as well be chopping it up. And so obviously this is evolved for that particular process. That's all I have. Good luck on Monday. Any questions come up, please feel free to email me.